Hey everybody, welcome back to Drawing with Mr. Hieggelke and Mr. Conroy. Today we are covering mighty mitosis. It's the one that makes multicellular organisms mature and replace worn out cells. And if you're a single cell organism like bacteria, paramecium, amoeba, then this is asexual reproduction. So we're going to start with the parent cell right here, and I'm going to use red and green. You can use whatever color you'd like, just make sure that they're very different. And we're going to draw a couple of chromosomes. One is going to be, for this conversation, from biological mom, and one is from biological dad. We're going to have biological mom shown in red, and so we're going to make one big chromosome. And we're going to make another smaller chromosome just to differentiate that we know that there are two different chromosomes. And then here is one from dad and a small one from dad. And the goal here is that we don't lose any chromosomes in the end, so that our daughter cells have the same amount of DNA as the parent cell. So here we go. We're going to start with prophase. In prophase, since we are going to get two new cells, we're going to have to double this DNA. And so what we're going to do is we're going to have the original, and the original is going to duplicate, and it comes out looking kind of like an X. But here's one on the left, here's the original, and then we've got a small one, and that's going to duplicate itself. So there's the new one. And then dad's. Here's dad's original, and the duplicate, and then the small one from dad. That duplicates also. I like that. All right, so prophase. In metaphase, meta means middle, and not that you have to remember that, but what's going to happen is the chromosomes are going to be brought to the middle of the cell, and so we're going to put, it can be in any order. So there's the big one from dad, big one from mom, and we'll do another one from dad, and another small one from mom. And the way this happens is there are these little, call them spindle fibers, that go out and attach in the middle where they're connected and kind of organize them into the center, kind of like that. So they were all jumbled around like up here, but then the spindle fibers bring them in the center. How they know where the center is, we have no idea, but they do it. And then what happens is the spindle fibers are going to reel their side over. So all the ones on the right side are going to be pulled this way, and all the ones on the left side will get pulled that way. So here's what we're going to do. We're going to have one big green one. So we're going to take this right one and put it over here. And we're going to move the left side over here. Same with mom. Here's mom's big one. And we're going to put that one over here. And the left one there. Like that. And then we're going to do the two small ones. So the right side will get pulled this way. So here's this one, and this one will get pulled over here. Then we're going to pull the small ones over as well. So this side goes this way, this side goes that way. Bada bing, bada boom. And now we've got two daughter cells from the parent. So we've got one big green, one big red, small green, small red, and if we take a look back up at the parent cell, we haven't lost any DNA. It's exactly the same. And that's mighty mitosis. All right, have fun.